So following on from the anatomy of the respiratory system, and this lecture is going to focus on the physiology. We're going to discuss the steps involved in breathing in and out, talk about the exchange of carbon dioxide and oxygen in the alveoli and the tissues, as well as how these gases are transported in the blood. To do that, we have another 5 learning objectives that we need to cover. Firstly, to describe the events involved in pulmonary ventilation, so breathing in and breathing out. To differentiate between different lung volumes and lung capacities. To describe the exchange of oxygen and carbon dioxide in external and internal respiration. Describe the transport of these gases in the blood. And then to describe how the body controls breathing. So getting straight into it, learning objective number 1, to describe the events involved in pulmonary ventilation. Now, before we get into pulmonary ventilation specifically, I want you to think more broadly about the function of the respiratory system. So the primary function of the respiratory system is gas exchange. We want to take oxygen from the atmosphere and deliver it to the cells of our body, and then take the carbon dioxide that we produce in those cells and then expel it back out into the atmosphere. This process of gas exchange is called respiration, and it occurs in 3 distinct steps. First, we have pulmonary ventilation, which is what we're going to talk about in this learning objective. Pulmonary ventilation, which you can also just call breathing, is the inhalation and exhalation of air, and it involves the exchange of air between the atmosphere, or the external environment, and the alveoli of the lungs. We then have external respiration, sometimes also called pulmonary respiration. This is the exchange of gases between the alveoli of the lungs and the blood in the pulmonary capillaries, or the capillaries which surround the alveoli. In this process, oxygen moves into the pulmonary capillaries and carbon dioxide moves out. The last step is internal respiration, sometimes also called tissue respiration. And this is the exchange of gases between systemic capillaries, so this is the capillaries that we find everywhere else throughout the body, and the tissue cells. So in this step, the oxygen moves from the blood to the tissue cells, and the carbon dioxide moves from the tissue cells back into the capillaries. So now focusing just on pulmonary ventilation to begin with, and in pulmonary ventilation or breathing, air flows between the atmosphere and the alveoli of the lungs because of alternating pressure differences created by changes in the volume of the thoracic cavity due to the contraction and relaxation of respiratory muscles. So very, very wordy, but we have air moving in and out of our lungs due to changes in pressure which are caused by changes in volume of the thoracic cavity when our respiratory muscles contract and relax. But before we go into the distinct steps that are involved in breathing in and out, there's 2 concepts that I want you to be familiar with. The first is that breathing begins with the contraction of our respiratory muscles. So the first step of breathing is actually the contraction of a muscle. The most important muscle for inhalation and exhalation is the diaphragm. And the diaphragm is this muscle here. So the diaphragm is a dome-shaped skeletal muscle that forms the floor of the thoracic cavity. It's skeletal muscle, so we do have voluntary control over it. When this muscle contracts, like when we inhale, it actually flattens out and moves down. It loses that dome shape. And when it flattens out and moves down, it increases the volume of the thoracic cavity. When the diaphragm relaxes, it then moves back upward and back into that dome shape. It reduces the space of the thoracic cavity. Now, contraction and relaxation of the diaphragm causes about 75% of the movement of air during normal quiet breathing. So what we mean by normal quiet breathing is just that breathing that you undertake at rest, just breathing in and out normally when you're not really thinking about it. So alongside the diaphragm, we also have other muscles that are involved in ventilation. We have our external intercostal muscles. So these are these muscles here that connect each of the ribs, and they sit on the external surface, so more superficial. When they contract, they elevate the ribs and kind of increase that thoracic cavity volume. Our external intercostal muscles work with the diaphragm during that normal quiet breathing. When we need to forcefully breathe in or breathe out, we also have accessory respiratory muscles, which help even further either increase or decrease the size of the thoracic cavity. So during inhalation, we have our sternocleidomastoid muscle, so muscles that run down either side of your neck, as well as your scalene muscles, which are these ones here. These two muscles will elevate the first two ribs, so again, increasing the size of the thoracic cavity. We also have our pec minor muscles, which are not pictured here, but would usually sit kind of in this location, and they elevate the third and fourth rib when we breathe in. During exhalation, so when you breathe really hard out, we also have our abdominal muscles and our internal intercostals. So our abdominal muscles, our rectus abdominis, our obliques, and our transverse abdominis will help put pressure on the abdominal region. Our internal intercostals, which sit kind of on the underneath of our external intercostals, will bring the ribs down and decrease the space in the thoracic cavity. So that was the first concept, that breathing begins with the contraction of our respiratory muscles. The second concept that I want you to be aware of and keep in the back of your mind is that air moves into or out of the lungs due to a change in intrapulmonary pressure. So intra means within, pulmonary is referring to the lungs. So here we're talking about the pressure within the lungs or within the alveoli. And this change in pressure is caused by a change in the volume of the thoracic cavity. Now, this relationship between the volume of a container and the pressure within it relates to the concept of Boyle's law. So I do want you to be aware of what Boyle's law is. So Boyle's law states that the pressure of a gas in a closed container is inversely proportional to the volume of that container. Now, what that means is that if the size of a closed container is increased, the pressure inside it will decrease. If the size of a container decreases, then the pressure within it will increase. So if we give you a bit of a visual to try and understand that concept, we've got a closed container here. The volume of that container is 1 litre, and apologies for the American spelling. The pressure of the gas molecules within that closed container is 1 atmosphere. If we now reduce the volume of that container, the pressure inside must increase. So we've reduced the volume by half and the pressure has doubled. So if volume increases, pressure decreases. If volume decreases, pressure increases. So now thinking about that concept specifically when we look at our lungs and our thoracic cavity, at rest, our thoracic cavity looks like this. Our diaphragm contracts and moves down. It increases the volume of our thoracic cavity. If the volume has increased, the pressure must decrease, and that causes air to rush into the lungs. We then have our diaphragm relax, so it moves back up into this dome position. The volume in our thoracic cavity is now decreased. So if the volume has decreased, the pressure must increase, and that increased pressure pushes the air out of our lungs. So thinking about those two concepts or principles, we're now going to list the steps or the events that are involved in inhalation and exhalation. So starting with breathing in, and just before each breath in or each inhalation, the pressure inside the lung is equal to the air pressure of the atmosphere. At sea level, this is about 760 millimetres of mercury or 1 atmosphere. For air to flow into the lungs, as we saw on the previous slide, the pressure inside the lungs or inside the alveoli must become lower than the pressure of the atmosphere. How we do this is by increasing the size of the lungs or increasing the volume of the thoracic cavity. So the first step of inhalation is that our inspiratory muscles contract. So during that normal quiet breathing, our diaphragm moves down, our external intercostals move the ribs up. As a result of this movement, the volume of our thoracic cavity increases. Because of the pleura that are attached to the thoracic cavity wall and our lungs, and that space between the pleura, which holds those two pleura together, when our thoracic cavity increases in volume, our lungs are stretched with the thoracic cavity and our intrapulmonary volume increases. So the volume within our lungs increases. Considering Boyle's law, if the volume in our lungs has increased, the pressure must decrease. It decreases to about 758 millimeters of mercury. Because air will flow from a region of high pressure to a region of low pressure, this drop in pressure within the lungs causes the air to flow down into the alveoli, and air will continue to move down into the lungs until no pressure difference exists between the inside of the lungs in the alveoli and the atmosphere or the external environment. So just quickly going over those steps again, and we will cover this again in the labs. But step 1 is that our inspiratory muscles contract. When these muscles contract, the thoracic cavity volume increases. Our lungs are stretched with our thoracic cavity, so the volume of our lungs also increases. Because the volume is increased, the pressure within our lungs must decrease. That change in pressure means that air flows from the atmosphere down into the lungs. So that's inhalation or breathing in. Moving on to exhalation, and breathing out works on the same principles as inhalation, but just in the opposite direction. So our inspiratory muscles relax. As the diaphragm relaxes, it moves back up into that dome position. The external intercostal muscles bring the ribs back down. With this movement of the muscles and the ribs, the thoracic cavity volume decreases. With the decreased volume of the thoracic cavity, the lungs, being elastic, will recoil, and the intrapulmonary volume, or the volume of the lungs, will decrease. Because that volume has decreased, we know that the pressure must increase. It increases again by about 2, so it goes up to 762 millimeters of mercury. This increased pressure within the lungs and the alveoli now causes the air to flow out of the lungs, moving from that area of high pressure within the lungs to low pressure outside of the lungs. And again, air will continue to flow out of the lungs until the pressure within the lungs matches that of the atmosphere. So once more for a recap, when we're breathing out, first step is that our inspiratory muscles must relax. This reduces the volume of our thoracic cavity. Our lungs will recoil and our intrapulmonary volume, or the volume of our lungs, will decrease. Because the volume decreases, the pressure will increase, and air flows out of the lungs until the pressure within the lungs and out of the lungs is equal. Now, there are a few factors that can affect pulmonary ventilation or breathing in and out that I just quickly wanted to mention here. These are the surface tension of the alveoli fluid. So I said in the previous lecture that our type 2 alveolar cells secrete an alveoli fluid. In that fluid is surfactant, which decreases surface tension. This stops the alveoli from caving in and sticking together at the end of each exhalation. If surface tension is high, the effort required to overcome this force and inflate the alveoli is high. If surface tension is lower, like when we have surfactant, the effort required to overcome this surface tension and inflate the alveoli is less. The second factor affecting our breathing in and breathing out is the compliance of the lungs. So this refers to how much effort is required to stretch the lungs and the chest wall. Excuse me, apologies. Now, high compliance means that the lungs and the chest wall will expand easily. Low compliance means that they will resist expansion, so they'll be a bit stiff. The compliance of the lungs is dependent on surface tension, like we just talked about, and the elasticity of our lung tissue. Normally, our lungs will have high compliance and they will expand really easy. You really don't have to think too much about breathing in and breathing out. However, decreased compliance is a common feature in many pulmonary conditions or lung disorders that scar the lung tissue itself so it becomes quite stiff, or in the case of premature babies who don't necessarily have enough surfactant. Lastly, we have airway resistance, which refers to the resistance of airways to airflow. So for example, as we're exercising and we're trying to expand our lungs during inhalation, our bronchioles will dilate. The larger the diameter of the bronchioles, the lower the airway resistance. The air can move much more easily through those passageways. So any condition which narrows the passageway of the air will increase airway resistance. An obvious example of this is asthma, where the bronchioles of the lungs will constrict. The narrower airways makes it harder to get the air in and also get the air out. And so breathing becomes much more laboured and much more effortful. Now moving on to learning objective number 2, now that we understand how we get the air into our lungs, I want to talk about how much air goes in and out of our lungs with each breath. And I want you to be able to describe and differentiate between the different lung volumes and lung capacities. So while at rest, a healthy adult breathes in and out about 12 times per minute. With each inhalation and exhalation, they move about 500 millilitres of air. The volume of one breath is what we call tidal volume. So you can see that here. This is one breath in, one breath out. Minute ventilation is the total volume of air that is inhaled and exhaled each minute. And it's calculated as respiratory rate, or how many times you breathe in and out in 1 minute, multiplied by this tidal volume. So let's say that the average person breathes in and out 12 times per minute. Their tidal volume is half a litre or 500 millilitres. Their minute ventilation would be 6 litres per minute. We breathe in and out 6 litres of air every minute. Now, the apparatus that we use to measure the volume of air that is inspired and expired is called a spirometer. A spirometer will record how much air goes in, how quickly air comes out, and it will give you a graph a little bit like this. So where we see the graph moving upwards, this is a breath in. Where we see the graph moving downwards, this is a breath out. Now we're going to have a go at using a spirometer in the labs, and I want as many of you to have a go as you can, because tidal volumes and some of our other lung volumes will vary considerably between one person to another, and even between the same person at different times. So I want you to have a bit of a think about why this might be the case. Now, we also have several other lung volumes that we can record using a spirometer, but all of these relate to a forceful breath. So not just a normal breath in and out. It's when you take a really big deep breath in and a really big deep breath out. So by taking a really deep breath in, you can inhale significantly more than the 500 millilitres that you do on a normal breath. This additional inhaled air is called your inspiratory reserve volume, and it's about 3,100 millilitres in males and 1,900 millilitres in females. Similarly, if you breathe out beyond just a normal breath out, you can get significantly more air out than our 500 millilitres. This volume is called our expiratory reserve volume, and is 1,200 millilitres in males and roughly 700 millilitres in females. Now, even after you have breathed out everything that you think you could possibly get out of your lungs, there is still a considerable volume of air that remains in your lungs. And this is to keep our alveoli partially inflated, as well as the air that will just stay within those non-collapsible airways. This is called our residual volume and is about 1,200 millilitres in males and 1,100 millilitres in females. So a recap of those volumes for you and just putting it all in words so you don't have to take too many notes. We have tidal volume, which is the volume of air that moves in and out of your lungs during normal breathing. We have inspiratory reserve volume, which is the additional amount of air that you can forcibly breathe in after a normal inhalation. We have the expiratory reserve volume, which is the additional amount of air that you can forcibly breathe out during a normal exhalation. And then lastly, after you've exhaled all of the air that you can possibly get out of your lungs, the air that remains is called our residual volume. So that was our lung volumes, and now we have our lung capacities. And our lung capacities are just the sum of two or more of these four lung volumes. So first up, we have our inspiratory capacity. This is all of the air breathed in after a normal breath out. So it's represented by this line here, and it's these two different lung volumes. So we calculate our inspiratory capacity by summing our tidal volume and our inspiratory reserve volume. We then have vital capacity, and this is all of the air that can be expelled from the lungs after a maximal breath in. So we can calculate that by summing our inspiratory reserve volume, our tidal volume, and our expiratory reserve volume. Our total lung capacity is the maximum volume of air that can be held within the lungs. So this is not moved in or moved out, but just the maximum amount of air that can fill your lungs. It's represented by this line here. And we simply have to sum the 1, 2, 3, 4 of our lung volumes. Lastly, we have our functional residual capacity. This is the air that remains in the lungs after a normal breath out. And this capacity is calculated by summing our expiratory reserve volume and our residual volume. So putting all of those into words for you, just as a bit of a recap, we have our inspiratory capacity, which is the volume of air that can be breathed in after a normal expiration. This is our tidal volume plus our inspiratory reserve volume. Our vital capacity is all the air that can be expelled after a maximal inhalation. So vital capacity is our inspiratory reserve volume, our tidal volume, plus our expiratory reserve volume. Our total lung capacity is the absolute maximum volume of air that can be held within our lungs. Total lung capacity is calculated by summing all of our four lung volumes. And then our functional residual capacity is the air that remains in the lungs after a normal expiration and is made up of our expiratory reserve volume and our residual volume. Now, the reason that we look at and we define all of these lung volumes and capacities is because based on a number of factors, so things like sex, height, age, smoking status, we can predict to reasonable accuracy what these values should look like. So then if we test a patient and their numbers come back significantly different to what we would expect, then it may be indicative of some kind of pulmonary or lung disorder. So we will go over this in more detail in the lab, but when you have a go with the spirometer, there are 3 values that we're going to focus on. We have forced vital capacity, or FVC, and this is the total volume of air that you can forcibly exhale after a maximal inhale. So you'll grab the spirometer, you'll set it all up, you'll take the biggest breath in that you possibly can, and then you'll blow out as hard and as fast as you can, trying to get all of the air out that you possibly can. We then have forced expiratory volume in 1 second, which we also term just FEV1. And this is the volume of air that's exhaled during the first second of this forced vital capacity test. Lastly, we have the ratio of FEV1 to FVC. And this is the percentage of air that's exhaled during the first second of our forced vital capacity test relative to the total volume of air that is exhaled. So when you do your big breath in and then big breath out, how much of that total amount of air that you breathed out was able to come out in that first second. So what you'll get from the spirometer is a number of values. You will also see a graph. And so normal typical results will look something like this. You have time along the x-axis. So this is the amount of time that the patient is blowing out for. Along the y-axis, we have volume, and this is the volume of air that they can expel. So for an average healthy person, the results look something like this. They should be able to expel a large amount of air in that first 1 second, and then a smaller amount of air for that remaining 5 or 6 seconds. What we are looking for is that this FEV1 to FVC ratio is about 80%. So what that means is that the person can get 80% of the total amount of air that they expel out in that first second. What we're then going to look at is the total amount of air that that person expels. And based on their age, size, smoking status, is that kind of normal or is that not? And I'm not going to go into any more detail about this right now, but in preparation for the lab, I want you to have a think about these values. Remember what they look like for a healthy person. And then have a think about what these values might look like for a person that has an obstructive lung disorder. So something like asthma, where your airways are obstructed or narrowed. And then have a think about what they might look like for a person that has a restrictive lung disorder. So this restricts or prevents the lungs from fully expanding. How might this graph differ? Moving on to learning objective number 3, and we have spoken about how we get air into and out of our lungs. We've talked about the different lung volumes and capacities. And now we're going to take a look at external and internal respiration, which is where we actually have that exchange of oxygen and carbon dioxide between alveoli and pulmonary capillaries, and then between systemic capillaries and tissue cells. So as a reminder, external respiration is the exchange of gases between the alveoli of the lungs and the blood in the pulmonary capillaries, or the capillaries which surround the alveoli. Internal respiration is the exchange of gases between systemic capillaries, which are the capillaries that we find everywhere else through the body, and the tissue cells. Now, the exchange of oxygen and carbon dioxide that occurs during external and internal respiration occurs via passive diffusion. And this diffusion is governed by the behaviour of gases as described by 2 laws. So there's 2 more laws that I want you to remember. So first, Dalton's law. And Dalton's law states that each gas in a mixture of gases exerts its own pressure as if there was no other gas present. Dalton's law is important for understanding how gases move down their pressure gradients during passive diffusion, and why we have the movement of oxygen, just as one example, from the alveoli air into the pulmonary capillaries, and then from the systemic capillaries into the tissue cells. Henry's law states that the quantity of a gas that will dissolve in a liquid is proportional to the partial pressure of the gas as well as its solubility. So this refers to how easily a gas will diffuse into blood, highlighting that the partial pressure of the gas, as well as how soluble it is or how easily it dissolves into a liquid, will affect how well oxygen and carbon dioxide move into and out of the blood. So first, talking about Dalton's law, which remember was that each gas in a mixture of gases exerts its own pressure as if no other gas were present. And the pressure of a specific gas in a mixture of gases is called its partial pressure. Now, the total pressure of a mixture of gases is calculated by summing all of the partial pressures of the gases in that mixture. So if we're trying to get the total pressure of this gas mixture, we simply have to sum the pressure of gas 1, the partial pressure of gas 2, and the partial pressure of gas 3, assuming there are 3 gases in this total mixture. Now, if we take atmospheric air as an example, and the air in the atmosphere is made up of a mixture of nitrogen, oxygen, argon, carbon dioxide, and water vapour, as well as a small amount of other gases. Atmospheric pressure is the sum of all of these partial pressures. Atmospheric pressure, which I've mentioned before, at sea level is 760 millimetres of mercury. Now, we can determine the partial pressure exerted by each gas in a mixture by multiplying the percentage of the gas in the mixture by the total pressure of the mixture. So I know that sounds a bit confusing, but I'll give you a few examples. So as an example, 78.6% of the gas in the atmosphere is nitrogen. If we multiply this percentage by atmospheric pressure, we get 597.4 millimetres of mercury. This is the partial pressure of nitrogen in the atmosphere. We'll look at another example. So oxygen, we have 20.9% oxygen in the atmosphere. We multiply this by the total atmospheric pressure. We get 158.8 millimetres of mercury, and this is the partial pressure of oxygen at sea level. We then want to make sure that it adds up to our total atmospheric pressure. If we sum all of these values, we get that atmospheric pressure of 760 millimetres of mercury. Now, don't stress too much about those equations. I'm not going to ask you to get a calculator out and do that maths. But what I want you to be aware of is that the partial pressures of a gas determine the direction of movement or the direction of diffusion of different gases. So both oxygen and carbon dioxide diffuse across permeable membranes from an area where its partial pressure is high to an area where its partial pressure is low. The greater the difference in partial pressure, the faster the rate of diffusion. So if we have a look at this image here, we have external respiration up the top. So remember, that's the exchange of gases between the alveoli in the lungs and the pulmonary capillaries, or the capillaries that wrap around the alveoli. We have internal respiration, which is the exchange of gases from our systemic capillaries into our tissue cells. So starting at the top, as our blood is coming back to the heart, coming out to the lungs, it's our deoxygenated blood, represented by the blue colour there. We have a low partial pressure of oxygen. So the partial pressure of oxygen is only 40 millimetres of mercury. In the alveoli, or the air within our alveoli, the partial pressure of oxygen is high. The partial pressure of oxygen is 105 millimetres of mercury. Now, because of this difference in partial pressures, the oxygen will diffuse from an area of high partial pressure to an area of low partial pressure, and it will move from the air in the alveoli into those pulmonary capillaries. Diffusion will continue in this direction until the partial pressure of oxygen is the same in the pulmonary capillaries as it is in the air in the alveoli. Now, while oxygen is diffusing from the alveoli into our pulmonary capillaries, carbon dioxide is diffusing in the opposite direction. So the partial pressure of oxygen in our deoxygenated blood is 45 millimetres of mercury. The partial pressure of the of the air within the alveoli is 40 millimeters of mercury. So not as great a difference, but it still is higher within the blood than it is within the air in the alveoli. What that means is that carbon dioxide goes from the pulmonary capillaries into the alveoli. And again, this direction of diffusion will continue to occur until there's no partial pressure difference between the pulmonary capillaries and the air in the alveoli. Now, if we come down the bottom and look at our internal respiration, so remember between our systemic capillaries and our tissue cells. So the blood has been oxygenated in our lungs. Oxygenated is showing the red, it's saying that there's lots of oxygen, goes to the left side of our heart where it's then pumped into the aorta and all around the body. So the oxygen content or the partial pressure of oxygen in our systemic capillaries is quite high. It's 100 millimeters of mercury. In our tissue cells, it's quite low. So it's 40 millimeters of mercury. And think about it, this makes sense. Our cells are using oxygen, so it's going to be lower within the cells than it is in the blood. Because of this difference in the partial pressure, the oxygen will go from the systemic capillaries into those tissue cells, and it will continue coming into the tissue cells until there's no difference in the partial pressure between the tissue cells and the systemic capillaries. Like with our external respiration, while oxygen is diffusing into the tissue cells, our carbon dioxide is going in the opposite direction. So here in our oxygenated blood, the partial pressure of carbon dioxide is low. It's 40 millimeters of mercury. In our tissues, it's high, so it's 45. Remember that we produce carbon dioxide when we break down substances to make ATP. So now the carbon dioxide goes from the area of high partial pressure to low partial pressure. It goes from the tissues into the systemic capillaries, so it can be taken back to the heart, then back to the lungs to be reoxygenated. Now, I just want to point out here that the partial pressures of oxygen and carbon dioxide that we have provided on this image are an example of partial pressures at rest. So, of course, when someone is exercising or being active, the oxygen requirements of the cells will increase, as does the production of carbon dioxide. Therefore, when we are exercising, the difference between the partial pressure of oxygen and carbon dioxide in the air in the alveoli and the pulmonary capillaries, and then between the blood in the systemic capillaries and the tissue cells will be even greater, and therefore diffusion will occur at a faster rate. Now, just coming back to those laws for a second, we're going to talk about Henry's law here, and this is relevant for both the diffusion of gases during respiration as well as the next learning objective that we're going to talk about. And this is how oxygen and carbon dioxide are transported in the blood. So remember that Henry's law states that the quantity of a gas that will dissolve in a liquid is proportional to the partial pressure of the gas and its solubility, or how easily it dissolves into a liquid. Therefore, when the partial pressure of a gas is high, the gas can more easily dissolve and stay in the body fluids. It also talks about the solubility of a gas. So, for example, carbon dioxide is dissolved in blood plasma much more easily than oxygen. It's 24 times more soluble in blood than oxygen. Similarly, even though we breathe in lots of nitrogen in that atmospheric air, this gas has no obvious effect on our body because at sea level, very little of it dissolves into the blood plasma because nitrogen has very low solubility. So how much that gas dissolves into the blood depends on the partial pressure and its solubility. So that brings us to learning objective number 4, which is to describe the transport of oxygen and carbon dioxide in the blood. So beginning with oxygen, and oxygen does not dissolve easily in water, as I just mentioned. Only about 1.5% of the oxygen that we breathe in is actually dissolved in blood plasma. About 98.5% of the oxygen in the blood needs to be bound to hemoglobin, which we find in red blood cells. So taking a look at hemoglobin, and that is this image that we've got on the slide here. And hemoglobin is a protein that is found within red blood cells. The heme, or the hemi portion of the hemoglobin, contains 4 atoms of iron, and each of these 4 atoms is capable of binding 1 molecule of oxygen. So each hemoglobin can take 4 molecules of oxygen. When oxygen and hemoglobin combine, they form oxyhemoglobin, which can easily be reversed, and we call it deoxyhemoglobin. Now, because only 1.5% of the oxygen in the blood can actually be dissolved in the blood plasma and then easily diffuse out into the tissue cells, that remaining 98.5% is trapped inside red blood cells. Because it's trapped, it's important to understand factors that either promote the binding of oxygen to hemoglobin, as well as what promotes the dissociation or the splitting or separation of oxygen from hemoglobin, so that it can then be released by the red blood cell and enter into the tissue cell. So that's what we're going to talk about here. And the first factor that influences the ability of oxygen to bind and dissociate or split from hemoglobin is the partial pressure of oxygen. The higher the partial pressure of oxygen, the more easily the oxygen will bind to the hemoglobin. Now, when hemoglobin has 4 oxygen molecules bound to it and it converts to oxyhemoglobin, we call that fully saturated because all 4 iron molecules are bound to an oxygen molecule. When the hemoglobin has less than 4 oxygen molecules bound to it, we say that it's partially saturated. So the relationship between the percent saturation of hemoglobin and the partial pressure of oxygen is illustrated in this diagram here. We call this the oxygen hemoglobin dissociation curve, so a bit of a mouthful. So we have partial pressure of oxygen down the bottom here. We have the percent saturation of hemoglobin here, and remember that that just means how many of those 4 iron molecules have oxygen molecules bound to them. And you'll note that at high partial pressures of oxygen, that hemoglobin is really well saturated. At low partial pressures of oxygen, the hemoglobin is less saturated. So what this means is that in pulmonary capillaries, where the partial pressure of oxygen is high, a lot of oxygen will bind to hemoglobin. In the systemic capillaries, near the tissue cells, where the partial pressure of oxygen is lower, less oxygen will bind to hemoglobin, which is good because more oxygen is released that can dissolve into the blood plasma and then diffuse into those tissue cells. So although partial pressure of oxygen is the most important factor that determines how much oxygen binds to hemoglobin, there are several other factors that will influence the affinity to which oxygen binds to hemoglobin, which just means how tightly that oxygen will bind to the hemoglobin. Now, when we look at each of these factors, they in effect shift this oxygen hemoglobin dissociation curve either to the left or to the right. So the left, moving this way, reflects a higher affinity of oxygen for hemoglobin, so it binds tighter. A shift to the right represents a lower affinity, so less oxygen will bind to that hemoglobin. Now, all of these graphs can be a little bit confusing, but what I want you to think is that the body is adjusting how it works so that the tissue cells continually get the oxygen that they need based on the conditions in which they're in. So if you keep in mind that metabolically active tissues need oxygen, they produce acids and heat as waste, all of this hopefully will make sense. So we're starting with acidity or pH, and as acidity increases or pH decreases, the affinity of hemoglobin for oxygen decreases, and oxygen dissociates more readily from hemoglobin, meaning that it can now diffuse into tissue cells. So the main acids produced by our metabolically active tissues are lactic acid and carbonic acid. When pH decreases, so it becomes more acidic, this oxygen hemoglobin dissociation curve moves to the right, meaning that at any given partial pressure of oxygen, less oxygen is bound to hemoglobin. It dissociates more readily. The opposite is also true. So if we have an increase in pH, a decrease in acidity, that curve will move to the right. So now the oxygen binds more readily to that hemoglobin. So the take-home message for this slide is that with a lower pH, or with more acids being produced, as what occurs when our cells are active, less oxygen is bound to hemoglobin. And this is a good thing because it means the oxygen can now diffuse into the tissue cells that need it. So low blood pH, more oxygen goes into the tissue cells. The next factor that affects the affinity of hemoglobin for oxygen is the partial pressure of carbon dioxide. And carbon dioxide can also bind to hemoglobin, but when it binds to hemoglobin, it changes the structure slightly so that now less oxygen can bind to that hemoglobin. So as the partial pressure of carbon dioxide increases, hemoglobin offloads that oxygen more readily. So that's this red line here. Our dissociation curve moves towards the right. So more carbon dioxide, or a higher partial pressure of carbon dioxide, more oxygen is offloaded that can diffuse into the plasma and then go into the tissue cells. Again, if you stop and think about it, it does make sense. If the partial pressure of carbon dioxide in the blood is high, the tissue cells must be increasingly active, requiring more oxygen to produce ATP. The high partial pressure of the carbon dioxide will reduce the affinity of hemoglobin for oxygen, so that less oxygen will bind to that hemoglobin and more is available to diffuse into the tissue cells. The next factor is temperature, and obviously within certain limits. Obviously the extremes are not going to be applicable here, but as temperature increases, so does the amount of oxygen released from the hemoglobin. So heat is a byproduct of metabolic reactions in our cells. Metabolically active cells require more oxygen and they release more acids and more heat. So again, just thinking about this logically, if tissue cells are active, they're producing more heat, they're going to require more oxygen. If these higher temperatures moves this dissociation curve to the right, it means less oxygen is bound to hemoglobin, so that more is available to diffuse into the tissue cells. Now, it's also interesting to look at the left side of this curve here as well when we're considering temperature. So in conditions like hypothermia, when you have a really low body temperature, our cellular metabolism will slow, the need for oxygen is reduced, and so more oxygen will stay bound to the hemoglobin because less is actually needed in the tissue cells. And then lastly, there are actually different types of hemoglobin that have different affinities for oxygen. And I just wanted to point this one out for some of our midwives or nursing students here. So as an example, fetal hemoglobin, so the hemoglobin found in a fetus in utero, has a higher affinity for oxygen than the adult maternal hemoglobin or the hemoglobin found in the mother. This means that when the partial pressure of oxygen is low, fetal hemoglobin can carry up to 30% more oxygen than the mother's hemoglobin. Now, as the blood goes from the mother into the placenta to go to the baby, it's already partially travelled around the body of the mother, so the partial pressure of oxygen can be quite low. So it's important that we have this shift of the curve to the left so that the fetal hemoglobin can actually bind more of that oxygen so that the fetus still receives oxygen that can then move into the tissue cells. Moving on to carbon dioxide, and carbon dioxide is transported in the blood in three main forms. 7% is dissolved in blood plasma, 23% is bound to hemoglobin, and 70% is transported as bicarbonate ions. Now, being transported either dissolved in blood plasma or bound to hemoglobin is pretty self-explanatory. However, how carbon dioxide is transported as bicarbonate ions is a little bit more complex. So as carbon dioxide diffuses into our systemic capillaries from the tissue cells, it reacts with water in the presence of a particular enzyme. This enzyme is called carbonic anhydrase, and it forms carbonic acid. The carbonic acid then dissociates or splits up into hydrogen ions and bicarbonate ions. Therefore, as more carbon dioxide enters the blood, more hydrogen ions and more bicarbonate will accumulate. Now, this is when we have carbon dioxide going from the tissue cells into the blood, but this equation also goes back the other way. So as blood passes through the pulmonary capillaries in the lungs, all of these equations are reversed. The bicarbonate combines with hydrogen, forms carbonic acid, and then we can breathe out water vapor and carbon dioxide. So now the amount of carbon dioxide that can be transported in the blood is influenced by the percent saturation of hemoglobin with oxygen, so how much oxygen is bound to that hemoglobin. The less oxygen that is bound to hemoglobin, the more carbon dioxide that can be carried in the blood. This relationship is known as the Haldane effect. I don't expect you to regurgitate that, but just in case you're doing some reading, that is what it's referring to. And now the last learning objective for the physiology of the respiratory system, and in fact the respiratory system overall, and that's to describe how the body controls breathing. So as we've already discussed, for breathing to occur, the size of the thorax is altered by the action of breathing muscles. Our muscles contract as a result of nerve impulses transmitted from respiratory centers in the brain, and then they relax when these nerve impulses stop. There are nerve impulses sent from clusters of neurons located in the brainstem. Collectively, they are called the respiratory center or respiratory centers, but they're located in two main regions and have two slightly different functions. We have the pontine respiratory group, which is located up here in the pons, and then the medullary respiratory center, which is located down in the medulla oblongata. So starting with our medullary respiratory center, and this is made up of two collections of neurons called the dorsal respiratory group, or DRG, and the ventral respiratory group, or VRG. Now, during normal quiet breathing, neurons of the dorsal respiratory group will send impulses to the diaphragm and the external intercostal muscles. These impulses start quite weakly, and then they increase in strength for about 2 seconds. They then stop altogether. So when the nerve impulse reaches the diaphragm and the external intercostal muscles, these muscles will contract. Apologies again, have a bit of a cold. Very topical for the respiratory system. Sorry. The diaphragm and the external intercostals contract for those 2 seconds. Those nerve impulses then stop, and the diaphragm and the external intercostals relax for about 3 seconds, allowing that passive recoil of the lungs and the thoracic cavity. The ventral respiratory group becomes activated when we have forceful breathing. So this is like when we're exercising, or maybe you're playing some kind of instrument that you need to blow into. During forceful inhalation, so forceful breathing in, nerve impulses from the dorsal respiratory group not only stimulate the diaphragm and the external intercostals, they also stimulate this ventral respiratory group to stimulate our accessory inspiratory muscles. So this is our scalene muscles, our sternocleidomastoid muscles, and our pec minor muscles. During forceful exhalation, our dorsal respiratory group is now completely inactive, but the ventral respiratory group will send muscles to our accessory expiratory muscles, so the muscles that help us forcefully exhale, which are our abdominal muscles and the internal intercostal muscles. So summarizing all of that in a bit of an image for you in case your brain works a bit better like this. So we have our dorsal respiratory group and our ventral respiratory group. So if we start here in the blue, which is during our normal quiet breathing, we get impulses from our dorsal respiratory group sent to our diaphragm and our external intercostal muscles. They cause those muscles to contract for about 2 seconds, and we have inhalation. We then have an absence of those nerve impulses, so the nerve impulses stop for about 3 seconds. Those muscles relax, and we have our normal exhalation. With forceful breathing, so that's what we've got in the yellow here, our dorsal respiratory group, again, will contract our diaphragm and our external intercostal muscles to help us forcefully breathe in, as well as stimulate our ventral respiratory group to stimulate those accessory inspiratory muscles. With our forceful exhalation, we have no input from the dorsal respiratory group, but our ventral respiratory group will stimulate or innervate the accessory expiratory muscles, so our abdominal muscles and our internal intercostals, and that causes our forceful exhalation. Our pontine respiratory group was then that collection of neurons in the pons. We know a little bit less about this pontine respiratory group, but what we believe occurs is that they transmit messages that modify the rhythm of breathing during things like exercise, speaking, and sleeping. So they play a role in both inhalation and exhalation. They actually stimulate both the dorsal respiratory group and the ventral respiratory group in the medulla, and they somehow modify the rhythm that we breathe in. Now, aside from the respiratory centers, breathing can also be modified in response to inputs from other brain regions and receptors in the peripheral nervous system. For example, the cerebral cortex has connections with the respiratory centers so that we can voluntarily alter our pattern of breathing. We can even choose to stop breathing for a short period of time. And this voluntary control is protective because it enables us to prevent water or gases from entering the lungs. And of course, that's gases that we don't want to enter our lungs. The ability to not breathe, however, even if we decide we don't want to breathe, we want to hold our breath, is limited by the buildup of carbon dioxide and of hydrogen ions in the body, which we'll talk about shortly. But when the partial pressure of carbon dioxide or hydrogen increases to a certain level, the dorsal respiratory group neurons of the medullary respiratory center are strongly stimulated. Those impulses are sent to the diaphragm and the external intercostal muscles, and breathing will resume whether or not you voluntarily decide to breathe. Nerve impulses from the hypothalamus and the limbic system can also stimulate the respiratory centers. And this is what causes things like emotional stimuli to alter breathing, such as what happens when we cry or when we laugh. Now, there's also sensory neurons that are responsive to chemicals, which are called chemoreceptors, which are very strong modifiers of breathing. So we have central chemoreceptors, which are located in or near the medulla oblongata in the central nervous system. They respond to changes in hydrogen ion concentration as well as the partial pressure of carbon dioxide. And it's the partial pressure of carbon dioxide and concentration of hydrogen ions in the cerebrospinal fluid, so the fluid that goes around the brain and around the spinal cord. We then have peripheral chemoreceptors. These are located in the arch of the aorta, as well as our common carotid vessels that run up either side of the neck. These are stimulated by, again, the hydrogen ion concentration in the blood, as well as the partial pressure of carbon dioxide and oxygen. But as I said, this time it's in the blood and not in the cerebrospinal fluid. Interestingly, the partial pressure of carbon dioxide is a bigger regulator of breathing than the partial pressure of oxygen. So if you're trying to adjust your breathing, it's easier to adjust how much carbon dioxide is in the blood rather than to adjust how much oxygen. Now that is it for the respiratory system. A bit of a long one, I apologise. Most of this, again, we will go over in the labs, but if you have any questions, make sure that you reach out. We will also see how all of this ties in with our next series of lectures, which is the cardiovascular system, and we can see how the respiratory system plays a strong role in the cardiovascular system. Thank you very much.